Okay, ladies and gentlemen, first of all, it's really nice to be here, and to be in Boston is fantastic. I tried to get here before, but they wouldn't let me in. But it's really nice to finally get here. It's in the evening, right? You've been working all day, so please, if you want to ask any questions, don't wait. Just stick your hand up or throw things at me, okay? And that way, hopefully, it'll be a little bit more of a dialogue than just sitting there listening, especially after you just had some food. So, as Brian mentioned, what I'm going to talk about, I'm going to talk about how the fact that ecology isn't rocket science, okay? And this is a quote by an Intertidal ecologist called Tony Underwood, and he often uses this quote when he's trying to talk about how we assess natural populations. And this is a group of school kids, this was yesterday, who are actually on the shores just around your lab. They're going out there, they're trying to assess natural populations. And I know that many of you have been trying to do that or doing that during some of your coursework. So this is really, if you like, a sort of warm-up act for Nova's talk coming up. And I'm going to go through some of the basics of how we assess populations. Many of this you may know, some of it you may not. Hopefully we'll come out with something at the end which is sort of interesting to everybody. When we're talking about the distribution of species, though, we're very interested, as Brian mentioned earlier, about how environmental conditions can influence the responses of species, but also their distributions. And we're really talking about different sort of scales. There's real large-scale biogeography, which is looking at distribution patterns over a large scale. We're interested because of climate change and these sort of things. And these can be affected as well by past historic events. So we've got large spatial, but also large temporal scales as well. And then we can get right down to smaller scales, like on this rocky shore here, and then down to, I think your pointer's dead, guys. Down to sort of microhabitat sort of areas where microhabitat scales where we've got very small scale changes in species distribution patterns. I think this is Brian's idea to try and get me to lose weight. Fantastic. I know. Easy, boy, easy. It's so exciting. When we're talking about patterns of distribution, though, we're talking about patterns of distribution of species, which we said, this idea of different scales is one of the things I want to get over today. And we've got scales, for example, these are bacteria growing in a plate. These are one of the pinnacles of evolution. These are marine snails. And then we can get all the way down to looking at large scales with elephants going over hectares. And the same basic principle occurs for any ecologist who's trying to measure the distribution of species, whatever scale we're actually working on. We need to be able to look at the abundance and distribution of species. And in order to do that, the reason we're trying to do that is that before we can actually try to understand why or how they will change, we have to understand these basic patterns. And so we cannot get to the processes until we've actually worked out the simple patterns of species distributions and abundances. And that's the way that Nova's going to develop more towards the process work now, later on, and then tomorrow we'll take that a little bit further on. So there is some sort of framework or path that we're going to try and follow in these talks. So the basic principle for ecologists is to start off, we need to know patterns of abundance and distribution of species. It's not just for ecologists, though. This is actually a real-world thing. I don't know, in the States, I think you have the Environmental Protection Agency. We have something called the EIA in Hong Kong, where we have to have an environmental impact assessment before we can develop any area. And that requires an assessment of the distribution and abundance of species. The one that's going on in Hong Kong at the moment is whether we should build a third runway or not. That's a multi-million Hong Kong and, of course, US dollar development, which is now being held back at the moment because of the Chinese white dolphin. Can they develop in that area? We need to know how it's going to affect the dolphins. And so we need to understand the distribution patterns of those animals. And so today I want to go through some of the ways we can do this. How can we assess the distribution and abundance of species? And the way I'm going to look at it is how can we do it well and how we can do it badly. And I'm mostly going to talk about badly and then try to, if you like, give you patches or how we can fix up and avoid doing it badly. So that's the idea of how I want to sort of wander through the talk. And I'm really going to talk about practical implications, going out and actually trying to measure the distribution of species practically. How do you go and do that? And when we talk about how we measure biological variables, there's a lot of ways we can try and do that. There's this way where we can actually take animals or plants or algae back into the laboratory where we can control all the conditions we work on. And a lot of people like to do this because you can sit in a nice air-conditioned office, you get to wear a white coat, and you get to control all these areas and all these variables, so you get this lovely, nice, clean data sets. These nice lines where very, very small variants around the data you've collected. And this really was one of the themes I want to talk about, because this is clearly a failed rocket scientist, okay? That's not the way you do it. This is the way the guys who can't handle it, that's what they reduce themselves to, is going back into the laboratory. The guys who can actually get out and do it, they're the guys who are out in the natural environment, okay? They're the guys who are out there dealing with natural variation, because populations of organisms living in the real world are variable. They're variable on spatial scales, and they're variable on temporal scales. And you have to deal with data sets like this. Believe me, this is a good data set, okay? I'd be proud of that. So you've got to deal with these different areas, and that's why I was saying that ecology is not rocket science, but actually to do it properly is really quite difficult. So that's what I want to try and follow through today. I'm going to use intertidal systems because they're cool, right? You're all here because you want to study marine biology. And intertidal systems are probably the best laboratory, best natural laboratory that you can actually find anywhere in the world. I'm just going to go through a few little pointers just to sort of bring us all up to speed so we're all on the same page. I'm sure that most of you know this already, but just a little sort of 101 crash course in intertidal ecology. As most of you know, the intertidal zone is defined by the rise and fall of the tides. There we go, rise, fall, rise, fall. And it's the gradient, the environmental gradient between being in the marine environment and being in the terrestrial environment. And according to where you are in the world, you can spend 2 times a day underwater, or in some parts of the world just 1 time a day underwater, and 2 times a day under air. And if you think about that, that's a very steep environmental gradient, because even in this room, I don't believe any of us have got gills, and so you can't cope with living under those conditions. But these organisms you've seen on the shore, you've been out measuring, they're able to do that. They're able to go underwater, and they're able to survive in air. The intertidal environment is also incredibly good to work on, especially rocky shores. Okay, none of this soft shore rubbish. Okay, we're talking about rocky shores now. Because organisms on rocky shores, they're actually stuck on the... I do that to wake people up, okay? So I'll be doing that a lot later on, okay? They're actually stuck on the rock surface, which makes them very easy to count, very easy to see, and very easy to quantify. So that's one of the areas that it's very useful for. It's a very dynamic environment, and it also has got a great diversity of life, so it attracts a lot of interest because of that. But from the point of view of what we're talking about, it's this environmental gradient, this strong vertical gradient caused by the rise and fall of the tides, which makes it an ideal place to look at the factors which affect the distribution and abundance of species. And this is the sort of thing which you can look at. This is, I think, something like 8 meters. I'm sorry, I don't know what that is in feet, okay? We use SI units as scientists. And then when the tide comes back in, it looks like this, okay? So that's basically the sort of 2 extremes of this gradient. And of course, it is a gradient. The tide comes in and the tide goes out. And what that creates is a gradient in immersion, which is being in air, and immersion, it almost sounds the same, right? Which is being in water. And so these are tidal curves on the left, and depending on what part of the world you live in, the animals on the top of the shore, they'll spend almost all their time in air. The ones at the bottom will spend almost all their time in water. And guess what? At mid-tide level, it's 50-50, right? And no matter where you are, you can identify, no matter how big the tidal range is, you'll still be able to identify this environmental gradient. And so because of this, what we find is that there's a lot of different environmental factors which change along this gradient. And Brian will be talking a lot about how we can measure many of these physical and chemical factors over the environmental gradient tomorrow night. So that's my advert for Brian's talk. But because of all these things, one of the nice things we see is that there is a very great change in the distribution of species over this vertical gradient. This is a shore in Hong Kong. The tidal range is 2.5 meters. So this is the bottom, and this is about 2.5 meters. So that's the entire tidal gradient. And what I hope you can see, although the lights are rather bright, well, you can see clearly there's a big black band running through the middle of this photograph, yeah? Any ideas what it is? Fantastic. Barnacles, good guess, but wrong. Muscles, fantastic. So it's a band of muscles. What do you notice about this band of muscles? Yeah, it's fantastic, right? There's no Photoshop here. This is real, okay? So these animals, they're only living in this very narrow part of this tidal gradient. And that's because of environmental conditions and the biotic interactions between them. So it makes it a great place for us to actually go and look at the distribution patterns of animals and algae and bacteria and even seagrasses if you need to. So that's what we're going to try to do today. I'm going to try and go through how we can do this well and how we can do it badly. And as I said, I'm probably going to err more on the side of how we can screw up, but then how we can fix it. Okay, so that's the sort of way I want to try and run the rest of the presentation, thinking about how we can make mistakes, but how we can actually fix those mistakes. So when we're trying to measure the distribution of these organisms, I think one of the first things we need to do is we need to make sure we adopt some sort of scientific principles. And I always try to think about this in really looking at 3 R's. Okay, the first one is representative. What we're trying to do is to get a representative estimate of the distribution and abundance of organisms. We're trying to get a value which is repeatable. We need to be able to transfer how we're able to measure the distribution and abundance of organisms, perhaps from one site to another site, or perhaps between different workers, so we all do it in the same way, and that means we can compare them. And also we need to do it in a way which is reliable or unbiased. And that's really important because we're not trying to make any decisions here about what we think the abundance of organisms are. We're scientists trying to give a factual account of the distribution and abundance of organisms and perhaps test for differences between different sites. And to do that, obviously we'll come to use some form of statistics. Now today, I'm only going to talk a little bit about statistics, just about distribution patterns. And I'm sure you're all familiar with the stats which I'm going to talk about. But it's very important to design the statistics you're going to use prior to doing any work at all. You need to know how to collect the data before you actually go out and do any data collection. And so later on, as you see, we get more complex. We need to make sure we know exactly what form of statistics we're going to use so that we can collect the data in the right manner that will allow us to use those stats. So that's how we're going to sort of wander through this today. I'm going to start with a case study. I'm going to talk about a case study about measuring the abundance of sea urchins in Cape D'Agula Marine Reserve. Now, you've all heard of Cape D'Agula Marine Reserve, right? No? Come on, Brian, what sort of job are you doing here? Okay, right. A little bit of it, okay? You can just, this is no test questions, okay? It's just a little bit of fun. So this is where you are. Does anyone know where Hong Kong is? It's over there, yes, fantastic. It's just about there, right? Okay, so that's okay. So Hong Kong's about here, right? Okay, so this is where Hong Kong is, and this is where your food came from. Okay? My God, it was still hot. That's amazing, right? That's incredible. Pretty cool. So this is where I'm from. Okay, I've been in Hong Kong for over 20 years, and please, if we have a chat afterwards, please nobody ask me if I speak Japanese, because that's where Japan is, okay? And this is where Wen Yun's from and her students. So this is China. China's quite big, okay? And tomorrow is China's National Day, okay? So we have to be nice tomorrow, okay? We've got to be nice to you tomorrow, so that's very important.